Lesson 2H, movement of very large molecules across the membrane. We have spoken about how small nonpolar molecules like oxygen, carbon dioxide, and ammonia can simply diffuse across the phospholipid bilayer, unobstructed due to the tiny pores or openings between the individual phospholipids. This is driven by a concentration gradient and KMT. Water may also diffuse across the membrane due to its slight polarity, but also in part due to its abundance, which creates a turgor pressure pushing water to move. However, water also benefits from channel proteins, which allow polar or charged ions that are small enough to simply flow through. This diffusion of water via the pores or channels has a special name called osmosis. There are also proteins that only transport specific molecules, which are gated, preventing the free flow of unintended molecules from moving across the membrane. These include the ligand, mechanical, and voltage-gated carriers that we spoke about in earlier lessons. We also spoke on a process of active transport, where protein pumps are used to push ions and other small particles from an area of low concentration to an area of high concentration using ATP energy. This is pumping particles against the concentration gradient. All of these methods are for the movement of small molecules or particles. The movement of large particles such as proteins or sugars require a larger opening than a protein channel or carrier can provide. To move large particles into a cell, a process called endocytosis occurs. Endo means in, while cyto means cell. This is the movement of material into the cell, where ATP energy is used to direct the cell's membrane to create a mouth-like shape to engulf the material it would like to take into the cell cytoplasm. When large solids are taken in by the creation of vacuoles, it's called phagocytosis. Phago is Latin for eating. Here we see an amoeba phagocytizing a paramecium, which will likely then fuse with vesicles filled with digestive enzymes called lysosomes once it's been taken into the cytoplasm. When a cell membrane conforms to bring in fluids using vesicles, it's called Pinocytosis, where Pino is Latin for drinking. This cell is bringing in fluids from its extracellular space, which will include water as well as dissolved minerals and nutrients. There is also a more specific form of endocytosis, in which the substrate that's to be brought into the cell first must be recognized and captured by protein receptors on the cell's membrane. Once these receptors are filled, it will cause a conformational change in the membrane, creating a vesicle, and the material will be brought into the cell. This process is called receptor-mediated endocytosis. In this photo, we see the receptors entirely coated, which causes the membrane to form a vesicle. Material can be brought into a cell, but material may also leave the cell. These items may be waste, but may also be products manufactured by the cell destined for other locations within the body, like hormones and neurotransmitters. The formation of membranous vesicles or vacuoles, often from the Golgi body, will fuse with the cell membrane, which will result in the contents being released into the extracellular space. This process is called exocytosis, where exo is Latin for outside. This table summarizes all the different transportation methods for moving items in and out of a cell. It identifies the method as being active or passive, as well as listing some materials using this transportation method in and out of the cell.